introduction, I'm excited to share some recent work that myself and members of my lab have been performing to understand antibiotic-resistant superbugs that we found on hospital surfaces in the United States and Pakistan. Although we most often hear about bacteria in negative context, the truth is microbial communities are essential for both health and disease in every environment that we've sampled on Earth. This includes things most pertinent to us as humans, like their roles in our guts, on our skins, and in our mouth. But really, every natural and artificial environment has a distinct composition of microbes. Today, we'll be focusing specifically on bacteria that are found on hospital surfaces. When bacteria get where they're not supposed to and cause infections, we rely on a large class of drugs called antibiotics to fix us. Antibiotics are small molecules that use the principle of selective toxicity to kill bacteria and fungi, but don't harm ourselves, and they don't work against viruses, which is why if you have a cold or flu, antibiotics are not going to help you. They're small molecules that specifically work by targeting cellular processes that bacteria need to survive. The main types of processes that antibiotics, again, are cell wall synthesis and cell membrane biogenesis. So the bacteria use these to keep bad molecules out and keep good molecules in. And antibiotics also work by stopping synthesis of the DNA, which bacteria need to replicate, and by stopping production of proteins, which bacteria need to grow and divide. Antibiotics were discovered simultaneously by accident in the mid-20th century by Alexander Fleming, who discovered penicillin, and Gerhard Domack, who discovered the sulfonamides. Penicillin is a natural product that's produced by a mold that Fleming found growing on a petri dish of bacteria. He noticed that bacteria weren't able to grow around the mold, and so he isolated the chemicals from that mold and very rapidly used penicillin in 1942 for clinical treatment. However, from analysis of stored away banked bacterial isolates, we actually know that clinical resistance to penicillin predates its use clinically and was first observed in 1929. Gerhard was using dye molecules from the textile industry in Germany to see if any of them could kill bacteria without harming humans. And he found a class of molecules called the sulfonamides that were able to do just that. Those entered clinical use in 1935, and we think because they're purely synthetic molecules that have never been exposed to bacteria in nature, resistance didn't develop until much later in 1939. Since those two instances, a large number of different antibiotic classes have been developed and then implemented clinically, as indicated by this graph. However, immediately following clinical deployment for all classes of antibiotics was the advent of antibiotic resistance. Compounding this development of antibiotic resistance is a 40-year gap in the production of new antibiotics, termed the innovation gap. Around the mid-1960s, society got very lazy and stopped investing in the production of new antibiotics, but bacteria were still gaining resistance. This innovation gap officially ended in the early 2000s with the advent of linezolid and daptomycin, which are great antibiotics, but they work against only a limited spectrum of bacteria, and I'll talk about why that's problematic at the end. Bacteria are able to gain resistance to antibiotics through a few different ways. One of the most common is through vertical transmission, where a progenitor bacteria is able to gain a resistance mutation in its DNA, and then through normal growth and division of that bacteria, is able to pass on that resistance-conferring mutation to its progeny. This takes time for the bacteria to grow and divide. So the real reason why antibiotic resistance is such a clinical threat is through horizontal gene transfer, where bacteria are able to actively share their DNA with other living bacteria. One of the most common ways is through transformation, where one bacteria is able to pick up naked DNA that's just sitting around in the environment. Another way is through transduction, where a virus that specifically infects bacteria is able to carry pieces of DNA from one bacteria into another. And the third way is a more active process called conjugation, where bacteria are able to share a portion of their DNA with another type of bacteria. Horizontal gene transfer leading to antibiotic resistance is already a huge clinical problem. It's attributed to 700,000 deaths in the United States in 2014, and current estimates based off of the development of horizontal gene transfer-based antibiotic resistance and the lack of new drugs indicate that the problem is only going to get much worse. By 2050, it's expected that 10 million deaths per year are going to be attributed to antibiotic resistance. And this burden of disease is going to disproportionately fall on developing countries in Africa and Asia because of the lack of healthcare infrastructure and ease of antibiotic access. Another context where we are really concerned about antibiotic-resistant bacterial infections is inside of the hospital. It's sadly ironic that one of the worst places for you to be when you're sick is in the hospital due to hospital-acquired infections. Hospitalized patients are especially vulnerable to bacterial infection, as many are medically immunocompromised or on some sort of mechanical augmentation. Accordingly, the human and financial toll exerted by hospital-acquired infections is a significant burden on our existing healthcare infrastructure. Bacteria in the hospital are able to go from a colonized patient to a susceptible patient via asymptomatic carriage on humans, which is why if you're in the hospital, it's essential to wash your hands in between different patient rooms. But they can also survive on a variety of inanimate surfaces, including medical devices and on furniture. Regular surveillance of the bacteria that live on these devices and furniture is a routine part of infection control measures at hospitals in the United States to stop the spread of hospital-acquired infections. But that's not the case in many hospitals in developing nations. This lack of surface bacteria surveillance is an immense threat to local patient safety, given that a meta-analysis identified a strong inverse relationship between the percent of infections caused by antibiotic-resistant bacteria and the gross national income of that nation. And although countries with gross national incomes comparable to the United States have a relatively lower percentage of infections caused by antibiotic-resistant bacteria, this map, which depicts the global dissemination of 4 different antibiotic resistance genes, shows that antibiotic resistance genes are not restricted by geography, which to us in the Dantas Lab is justification for studying bacteria that come from developing nations with a naturally high burden of antibiotic resistance genes. A previous graduate student in the Dantas Lab, Mitch Paseski, did some analysis to show that Pakistan is one such country with a naturally high burden of antibiotic resistance genes. He sequenced the DNA of over 160 antibiotic-resistant superbugs found from clinical specimens in the United States and Pakistan, and then he computed how similar these bacteria are to one another. For one such species, Klebsiella pneumoniae, he actually found that there was very little genetic difference from bacteria that were obtained in either the United States or Pakistan. But despite this overall similarity, when he looked at their resistance genes to see what was giving them this superbug phenotype, their resistance genes were completely dependent on if they came from the United States or from Pakistan, contrary to what you saw here. When I joined the lab, I was really interested in following up on Mitch's work and seeing if this result held true for bacteria that were living on their hospital surfaces, not just that came from patient infections. To test this, we devised a year-long longitudinal analysis of 4 ICU rooms at a tertiary care hospital in Rawalpindi, Pakistan, and then an equivalently sized hospital in the United States. Surfaces in these ICU rooms were initially sampled every other week for the first 3 months of the study, and then at 6 months and 1 year. We hypothesized that a high burden of superbugs exist on the Pakistan hospital surface, but a limited number of related superbugs exist on the United States hospital surface. Both study sites, over time, had 5 different high-contact surfaces swabbed in triplicate. One swab was saved so that we could get an idea of the entire repertoire of bacteria present on those surfaces. One was used specifically to grow the superbug Clostridium difficile, which is very sensitive to oxygen and will die upon exposure. And the third swab was used to inoculate a bunch of different petri dishes so that we can selectively grow different types of antibiotic superbugs that are important clinically. And we can then assign an initial species ID using clinical tools like this acronym MALDI-TOF, and then perform sequencing to get an idea of the entire sequence of A, C, Gs, and Ts, which make up these organisms' DNA. This is an example of all the petri dishes and tubes needed for a single time point. And as you can infer from this, it was a very laborious procedure. After 1 year of sampling in the US ICU, we were pretty happy to find a very small number of bacteria, only 6 total isolates that came from 2 different species, Escherichia coli and Acinetobacter baumannii, both of which are well-known hospital-acquired infections. But we were much more surprised to learn that a larger number of isolates and species exist on the Pakistan hospital surface. We found close to 300 total bacteria from at least 30 different species. Very interesting to us, though, is that Acinetobacter baumannii was the single most abundant organism in Pakistan and in the United States. It was also interesting to us that using this clinical tool, 7 bacteria isolates were not able to have a species ID assigned to them, which was our first indication that they could be a completely new species to science. So I then, similar to Mitch, got the entire sequence of A, C, Gs, and Ts, which make up these organisms' DNA, and then I computed how similar all of those bacteria are to one another to construct this heat map. From a comparison of the blocks in this heat map to what our clinical tool called the bacteria, we were able to figure out what we thought was really 30 species was actually 36 existing species and 11 completely new species. It was very clear, though, that although we found a bunch of these interesting new species, the most abundant organisms that we found on the Pakistan hospital surface were the different antibiotic-resistant superbugs that the CDC is very concerned about, and in 2013 ranked in a resistance report either as serious or urgent antimicrobial resistance threats. Using this approach, we were also able to figure out what those 7 bacterial isolates that were initially unassigned really were. 2 of them were very interesting to us because when we played them on a bunch of different augers, this bacteria resembled another serious antimicrobial resistance threat, extended spectrum beta-lactamase producing Enterobacteriaceae. And so we were really surprised that it wasn't one of the recognized species because these are such important human pathogens. We isolated this organism. We turned strains BP1 and BP2, and then did electron microscopy to see what this bug looks like. It was very apparent to us that it's a rod-shaped bacteria that can sometimes exist in these clusters with an interesting looking protein linker between them. We did more testing to see what other antibiotics this bacteria is resistant to, and we were shocked to learn that it is technically multi-drug resistant because it's resistant to 4 different classes of antibiotics. And when we looked more in depth in the genome of this bacteria, we found 18 total antibiotic resistance genes, which does indeed make it a very serious antimicrobial resistance threat. So although we figured out that this bacteria could be very important for human health, we didn't still have a good idea of what it was. So using another metric of similarity between different bacteria's DNA, I figured out that our 2 strains, BP1 and BP2, were in a family of bacteria called the Enterobacteriaceae. And when I took different bacteria from within this family and computed how similar our 2 strains were to them, we saw that on this phylogenetic tree, they're a very clear branch away from the existing bacteria, which to us is justification for not just being a new species, but actually a completely new genus of bacteria. When we zoomed in on the genome of these 2 strains, we saw evidence of horizontal gene transfer because 3 different antibiotic resistance genes, shown here in orange, green, yellow, were very close to a gene involved in horizontal gene transfer in purple. We're not able to say if this bacteria acquired this sequence from another bacteria or if it is able to have the capacity to give it, but it's evidence that some sort of gene exchange may have been occurring. The end result of this is we wrote a paper which delineates these 2 strains as a new genus called Superbicebacter electus, which is Latin for a resistant bacteria derived from a surface. BP1, E. coli, and a lot of other bacterial superbugs are gram-negative. Bacteria can really be broken down into gram-negative and gram-positive, the major difference between them is that gram-negative bacteria have a large outer membrane, which is absent in gram-positive bacteria. This outer membrane is a huge barrier to getting antibiotics inside of the bacteria. I mentioned earlier that linezolid and daptomycin are good antibiotics, but they only work against gram-positive bacteria because they're not able to pass the outer membrane in gram-negative bacteria. As an example of how youth can get involved in the fight against antibiotic-resistant bacteria, Vinay was very interested when he joined our lab in developing ways to restore the efficacy of antibiotics against gram-negative bacteria. To do this, he devised a fluorometric assay that used a small molecule which produces a fluorescent signal once it gets inside of the bacteria, but not outside. He then mixed this fluorescent molecule with the pathogen E. coli and different antibiotics, colistin, ampicillin, and kanamycin, to show that only one of these antibiotics, colistin, was able to allow the fluorescent molecule to enter E. coli, much better than ampicillin or kanamycin. This holds promise for using colistin as a partner with other antibiotics to get drugs into gram-negative bacteria so that they can then stop these infections. And with that, I'd like to thank all members of my lab, and especially my boss, Gautam Dantas, for their insightful discussions and support, and a large number of collaborators and funding sources. And thank you for your time, too.